It's my honor to introduce Dr. Marcelo Bonini, who is a professor in the Department of Medicine at Northwestern University at the Feinberg School of Medicine. He's also Associate Director of Education and Training at the Robert H. Luria Comprehensive Cancer Center. Dr. Bonini received his Bachelor of Science in Chemistry from the University of Sao Paulo in Brazil in 1999, and then stayed at the same institution where he did a Ph.D. in biochemistry and received his Ph.D. in 2004. During that time, he published 10 papers, including 4 as first author. He moved to Research Triangle, where he continued his training in redox biology as a postdoctoral fellow at NIEHS from 2004 to 2009, working with Dr. Ronald Mason. During this time, he received a number of Young Investigator Awards, published 17 papers, and won an NIEHS Intramural Paper of the Year Award in 2008. All this work established his expertise in redox biology. He was recruited to the University of Illinois College of Medicine in Chicago as an assistant professor and director of the Oxidative Stress Core Facility in 2009. He was promoted and tenured to associate professor in 2015, and then was recruited away to Milwaukee to the College of Medicine of Wisconsin in 2018, where he won an award for excellence in research and was promoted to professor in 2015. He then was recruited to Northwestern in 2020. He's published over 100 peer-reviewed research papers, 16 reviews, 3 book chapters, holds 1 patent, and has 2 provisional patents. He serves as an editor for PLOS ONE and for Frontiers Oxidant Physiology, and is on 2 journal editorial boards. As if this didn't keep him busy enough, he has served as program director and chair for national scientific organizations, including as chair for diversity, equity, and inclusion for FASEB. He served on numerous national grant review panels and currently is chair of the NIH Systemic Injury by Environmental Exposures, SIEE study section. He has trained 7 postdocs and 1 PhD student and has co-mentored 13 trainees and is currently mentoring 3 postdocs. He is very well funded. He's PI on the P30 Cancer Research Center grant in its 27th year at Northwestern, and he's PI on 3 NIH R01s funded by NCI, NIEHS, and NIAD. His research on how environmental exposures to heavy metals, including arsenic and cadmium, changed the redox status in the nucleus, driving carcinogenesis, his examination of nitric oxide and reactive oxygen species and macrophage functions, and his work in cancer cell signaling and metabolism clearly is of interest to those in this room who are members of CIEHS and those in the audience online today. So, Dr. Bonini, we look forward to hearing about your research.
So good morning, everyone, and thank you so much, Carrie, for the introduction. Thank you also for hosting. Thank you, Chris, and thank you, John. It's great to see you guys. I was telling people earlier that it's wonderful. No. It's really wonderful to be doing this again after almost 2 years. I actually have seen little squares on a Zoom screen. I never thought that it would be at the same time amazing and intimidating to see people present again, but it's really awesome that this experience is happening in Louisville, a place that I have so many friends for so long. So today we are going to talk about a lot of things and nothing specifically. There are 3 short stories, and there is slices of the research program that has started at MCW a couple of years ago and now has come to fruition at Northwestern, where we are trying to understand how changes to the electrochemical environment of the nucleus modifies transcription, and in doing so, modify cellular phenotypes. And what I mean with that? Well, transcription factors are proteins. So if they come to the nucleus and it's more oxidizing, those proteins might be oxidized and partner with different co-regulators, co-activators, co-repressors, and that may change transcription. That may change DNA folding, and if there are different genes that are being exposed, that also may change transcription. And finally, if you have an environment that is oxidizing link RNAs or molecules that regulate interactions in the nucleus, that also may affect transcription that way. Though the redox state of the cell and the cytoplasm has been studied for many, many years, there are thousands of papers that shows how reactive oxygen species alter signal transduction in the nucleus, it's much less clear what happens in the nucleus. So here are some pathways that are featured. For instance, how oxidants altered thiooxidation in P10, and that makes AKT more active. That's very well described. Or how transcription factors like NRF2 and HIF1 are activated by reactive oxygen species in the cytosol. But there are enough papers and reviews in the topic that we can talk about on a different occasion. It's known, however, from many studies of DNA damage caused by oxidants, that oxidants reach the nucleus and have activities there. A lot of the study in this field has focused on how carbonate radicals and nitrogen dioxide and hydroxyl radicals and other oxidants damage DNA directly by altering the bases that compose DNA or by causing strand breaks. Much less is known about how reactive oxygen species modify transcription factor activity, as I alluded to, or transcription factor binding to specific targets, gene targets in the chromatin, or how it affects transcription factor release, or how it changes the structure of chromatin itself. So the three short stories that come after, that we start after this slide, focus on each one of these potential aspects. The first one, how we figure out how reactive oxygen species change transcription factor recognition of different targets, therefore changing the function from pro-inflammatory to anti-inflammatory. On a second story, I'm going to talk about how a modification of a mitochondrial protein that is normally an antioxidant defense makes the protein a nuclear demethylase protein that now regulates stemness gene expression. And in the last piece of the talk, I'm going to focus on some studies that we have showing that changes to the redox state of the nucleus, the electrochemical state of the nucleus, changes chromatin arrangement, opens chromatin, and allows cells to explore a much larger range of the genomic landscape that is available to the cell. So the concept here is that the redox state of the nucleus is really a very powerful, underappreciated force regulating transcription at multiple levels, from chromatin organization to transcription factor function and association with partners. So to be able to talk about the first story, I have to introduce NF-kappa B and the pro-inflammatory signaling cascade. NF-kappa B is a master regulator of inflammation. We have found in 2015 that when, and I think that the next slide is better, when NF-kappa B is activated, that can happen because receptors are activated by cytokines, pro-inflammatory cytokines, or bacterial products such as LPS. A number of downstream signal mediators get activated, but all of them converge to the phosphorylation of I-kappa B, or inhibitor of kappa B. And I-kappa B, when it's phosphorylated, it's degraded, and it releases two subunits, P65 and P50. In their free form, P65 and P50 go to the nucleus, and then some very interesting magic happens there. They can either become heterodimers, or P50 and P65, and that's your NF-kappa B1 transcription factor that is a pro-inflammatory molecule, or P50 can dimerize with itself, making the P50 homodimer, and that is anti-inflammatory in nature. And we had figured out that the nitrosation of a protein called suppressor of cytokine signaling 1, that is published in JAM, it regulates whether P65 is ubiquitinated and degraded in the nucleus, exported and degraded, therefore increasing the availability of P50 to dimerize with itself, or if SOX is not, basically if it's not nitrosated, it quenches P65. If it's nitrosated, P65 is free to bind P50, and you have the pro-inflammatory configuration. So what we started thinking is that production of nitric oxide in the nucleus by an enzyme called nitric oxide synthase 1 would then regulate whether you have pro-inflammatory configuration or anti-inflammatory configuration. Studies to explore that show that that's actually the case when we use macrophages prepared from bone marrow of wild-type animals, so they have the ability to produce nitric oxide in the nucleus. You see that activation with LPS makes the P65 subunit go to the nucleus, that's expected. P50 also goes to the nucleus, that's also expected. But while P65 is stable in the nucleus of the wild type, it doesn't really accumulate in the nucleus of the NOS1 knockouts that do not have the ability to produce NO, leading to an accumulation of the P50 form. If we quantify the amount of P65 and P50 in the nucleus in these two types of cells over time, you see that there is an increase in P65 and P50. In the case of the knockout, you see that actually P65 is much more abundant in the nucleus, and if you calculate the ratio, you see that there is a more favorable P65, P50 environment in the wild type than the NOS1. We also did a functional assay, which is the EMSA assay, it's electromobility shift assay, and we used oligos that are either recognized by P60, P50, P65, P50 heterodimer or the P50 homodimer. And what you see is that in the wild type macrophages upon activation, you have more of the heterodimer, the pro-inflammatory heterodimer, and less of the anti-inflammatory homodimer, and this relationship changes in the NOS1 knockout. So technically, our hypothesis that nitric oxide regulates transcription factor configuration held, and you should expect a downregulation of the pro-inflammatory gene transcription with an increase in the anti-inflammatory genes. However, what we saw was very interesting because although the hypothesis was correct, it depended on specific genes. For instance, IL-1 beta is a pro-inflammatory cytokine. You see that it increases in the wild type after activation with LPS, and that's what is expected. However, it's lessened in the NOS1 knockout. Interferon beta is another pro-inflammatory cytokine, and you see that it increases in the wild type and much less in the NOS1. So you see a suppression of pro-inflammatory cytokines. However, when you look at CXCL10, which is a pro-inflammatory chemokine, you don't see any difference. So although we have an NO-dependent regulation of configuration, it affects different gene targets differently. And that was very interesting because we expected an across-the-board downregulation of these pro-inflammatory gene targets, but we actually found there are at least two patterns. Pattern 1, which is dependent on redox regulation, and no regulation in this case, and pattern 2, which is basically NO-insensitive. And this is just a Western blot to show you that nitric oxide synthase 1, which is the largest source of NO in the macrophage, is indeed in the nuclear fraction. So we started thinking that maybe what this was doing was impacting the binding of P65 to the different gene targets. And then we decided to do a cheap PCR assay focusing on pattern 1 and pattern 2 genes that we had identified. And what you basically see here is that IL-1 beta, interferon beta, and IL-12, all pro-inflammatory genes, as expected, as known, P65 would be recruited to the promoter sites of those genes after activation with LPS in the wild type, but interestingly, not in the NOS1 knockouts. Same was true for interferon beta, and same was true for IL-12. However, and of course, that the recruitment of P65 to the promoter regions of these pro-inflammatory genes would be reduced in the NOS1. That was also what we expected. However, when you look at CXCL10, pattern 2 gene, you see that recruitment of P65 to the promoter region was basically the same, either in the wild type or in the knockout. So that established that there is a group of genes that is impacted by redox regulation in the nucleus and a group of genes that is not. And that was new and interesting. Now the question became why. So we knew that SOX1 bound P65 that was actually bound to DNA. There was a paper that showed in Science 10 years before our paper showed in JEM. So we had a different hypothesis now, getting the two concepts together, that what's really happening is that SOX1 is not just floating in the nucleus waiting to trap P65, but it's actually it's a lid that actually fits the span and regulates in a promoter gene-dependent manner where P65 can bind and where it cannot. And that nitric oxide would be this push that would displace SOX, allowing P65, P50 to bind there. So what we hypothesized is that in a condition where and NOS1 is not active, nitric oxide is not produced, SOX1 sits on specific promoter regions thereby ubiquitinating P65 that approaches those promoters. Those would be our pattern 1 genes that require SOX to be nitrosated, displaced, and then allow these genes to be transcribed. That also allows inflammation to be activated very quickly because you just need a pulse of NO and SOX is displaced while maintaining these genes available for transcription at all times. So the next experiment that we did, we just went back and did a very difficult experiment, was the cheap PCR of SOX1 on those genes, same genes. For those that have worked with SOX1, the antibodies are not great, so chipping it is not easy. So what we saw is that, as expected, in the wild type BMDMs, we saw a reduction, an NO-dependent reduction in the SOX1 binding to the promoter regions of IL-1 beta, again, interferon beta, and again, IL-12. But when we look at CXCL10, SOX doesn't change. It's either not there or it's not moved from that site by nitroxide. So that basically established a group of genes that require nitric oxide to displace that SOX, that temper that is on the gene, so P65, P50 can't go there. And how does that affect the phenotype of these macrophages? And I'm just going to show the main data about each one of these projects. There's a lot more there, but we're talking about the redoxicity of the nucleus affecting cellular phenotypes. We decided to do an RNA-seq on these macrophages, on these BMDMs, both wild type and NOS1, and what we saw is that, not surprisingly, when you activate wild-type macrophages with LPS, you see the manifestation of a very powerful pro-inflammatory activity that is centered around interferon signaling. But when you use the NOS1 knockout, what you see is more of an anti-inflammatory phenotype that is centered around IL-4. That's really not surprising. So this mechanism is regulating whether macrophages become more pro-inflammatory or less. We also did flow cytometry using membrane markers that denote a more pro-inflammatory phenotype or less. And what you see is that when you activate this lung alveolar macrophages now with LPS, you see that there is twice as more pro-inflammatory macrophages in the wild type and half of the population of pro-inflammatory macrophages there's more pro-inflammatory in the wild type, half in the NOS1 knockout, and that reverses when you look at pro-inflammatory macrophages. And how does that impact physiology? Well, basically, we actually did experiments where we collected now the plasma of mice that received LPS injection 8 hours after the injection, and what we see is that in the plasma there is an increase in pattern 1 pro-inflammatory cytokines in wild type. That increase is much lessened in wild type, and it's rescued with NO. So when you give LPS and nitric oxide, now you see that those pattern 1 genes get turned on just like they are in the wild type. So indicating that this mechanism does regulate the amount of pro-inflammatory cytokines that you get from a very powerful pro-inflammatory insult into these mice. Interestingly, when you look at lungs from these mice, wild-type lungs have profoundly remodeled lung. This is actually normal. It's one of the reasons why septic patients, critically ill patients, die, because the vessels start to get leaky. There is a reduction in the airway space, so oxygenation levels go down. You see that when we stimulate systemic inflammatory insult in the mice with LPS, IP, that happens with lung remodeling here. When we use NOS1 knockout and give LPS, we see that very little happens. The structure of the lung remains the same. There's no infiltration of immune cells and no leaky lungs, no edema, unless we give LPS and SNAP, which is an NO donor, and that reconstitute that pro-inflammatory activity in the lung. So this basically indicates that the manifestation of the pro-inflammatory phenotype of macrophages requires a change in the nuclear electrochemical environment that is caused by a production of nitric oxide. So what are the implications of this? So the follow-up project that we developed based on this is now studying the impact of aging, changing the redoxy state of the nucleus. We hypothesize that mitochondrial dysfunction that accumulates through life increases the oxidants in the nucleus. Oxidants would also displace SOX1 from those genes, and you have a propensity to have a much more robust pro-inflammatory response in the elderly when they're affected by viral infections or by whatever inflammation than young people because of this change of the electrochemical state of the nucleus. That's what the lab is focusing on now. So switching gears to the second story, I'm not going to tell you how a mitochondrial protein that is an antioxidant, its day job is being an antioxidant mitochondria, very important, well-studied one, and how the modification of this enzyme and the binding of a different cofactor changes it to becoming a nuclear histone demethylase. Before I do that, I'm going to tell you a little bit about manganese superoxide dismutase. It is a tetramer. This tetrameric protein binds manganese. It's its canonical cofactor. And in mitochondria, what it does is that it dismutates superoxide radicals that emerge from the electron transport chain into hydrogen peroxide and oxygen. However, we had found, and this is published in a number of papers from 2019 and 2017, that when metabolic dysfunction ensues and superoxide dismutase is now acetylated, it becomes a monomer, and the acetylated monomer actually binds iron. And interestingly enough, we started to find that this monomer would locate to the nucleus, and it became very important to us to know what it was doing there. In 2017, a new peroxidase activity was described for superoxide dismutase, meaning that it can use hydrogen peroxide to oxidize targets. And that was linked to the iron-bound form of SOD. So actually it became a matter of untangling all of this information into and getting into a cohesive story. So what we did is that first we labeled superoxide dismutase, and we made mutants that would either mimic acetylation. So when you replace the lysine residue by a glutamine, that basically mimics acetylation, or we remove the lysine and replace it with an arginine that cannot be acetylated. So SOD2 that is either acetylated, mimetic, or that cannot be acetylated. And then we overexpressed also the wild type form. So you see that the GFP labeled SOD, when it's expressed in cells in its wild type form, localizes vastly to the mitochondria. Here you see TMRM, which is a potentiometric mitochondrial indicator. When we use the acetyl-resistant form of the enzyme, you see that it's also vastly mitochondrial. Interestingly, the mitochondrial potential is a little reduced, but you still have mitochondrial activity here. When we express the Q form, the acetylmimetic, though, you see that, interestingly, it now distributes everywhere in the cell, inclusive of the nucleus, and you see only the nucleoli excluded here that are basically these black dots. So lots of experiments later, fractionation, we figured out that it was actually in the nucleus. This was not artifact from imaging. We looked at human samples. We found that in some tumors, SOD was actually in the nucleus of a rare population of cells. All of that done, it became very important to discover what SOD was doing in the nucleus. So one way of doing that is to look at partners and to look at the proteomics of the nucleus and see what was changing. And what was really surprising, at the time we had the option of doing the proteomics and the epiproteome, and we found that the tremendous changes in the methylation of histones, particularly in histone 3 lysine 9. So these were MCF7s that either expressed SOD, NLS SOD, or not, stimulated with doxycycline. And when we compare the proteome of cells that had SOD in the nucleus with regular cells, we found a reduction in the methylation of K9 and a reduction in the methylation of K27, histone 3. And if you guys work in epigenetics, you know that K9 trimethylation and K27 trimethylations are repressive marks. They repress transcription of the genes that are associated, whose promoters are associated with these marks. This is just a Western blot validating this finding. We have MCF7 cells and MCF7 cells that expressed SOD in the nucleus. When we looked at levels of histone 3, K9, 3-methyl, we found that they were reduced in the cells that had SOD in the nucleus, K27 the same. But when we looked at H3, K4, 3-methyl, which is a mark that normally denotes activation of gene expression, we saw no difference, indicating that SOD in the nucleus was acting as a demethylase of repressive marks that suppress gene expression. What we did to make sure that that was true, we then got SOD that we produced in vitro, either reconstituted with iron, the peroxidase, the demethylase, or manganese, the regular antioxidant. And we incubated that enzyme in vitro with calf times chromatin, and then repeated the Western blot. You see that only the SOD that was reconstituted with iron was able to demethylate that mark. If we used catalase that destroys hydrogen peroxide, you see that there's no longer the ability to demethylate, and MNSOD doesn't do that. This is just a quantification. We also know that in the process of demethylating histones, formaldehyde is produced. So we did the same assay, but we measured formaldehyde evolution. You see that but this used a little bit to change the experiment. We didn't use enzyme expressing bacteria, but we actually immunopurified enzyme from the nucleus of this MCF7 cells, or MCF7 cells that express the wild type, so vastly mitochondrial. And you see that only the SOD that was purified from the nucleus had this activity of producing formaldehyde when incubated with calf times chromatin, indicating that's the form that has the methylase activity. We also did ICP-MS, and we found out that this is also the form that is labeled with iron in vivo. But the big question here is, okay, now you have this enzyme that is normally mitochondrial that goes to the nucleus that now enables the utilization of hydrogen peroxide in the nucleus to demethylate genes. What does it mean for the biology of these cells? So we got the cells and we placed them in agar. That's a clonogenic colony forming assay. It's a good indication of cells that can actually form tumors when implanted in vivo. You see that cells that expressed wild type SOD didn't grow many colonies. You see a few here and there. NLS cells were able to grow many colonies. In terms of invasion, you see that the cells that have either nuclear excluded SOD or wild type overexpression, they don't invade a tissue. And what we did is that we placed them on top of Matrigel, and then we attract them to a cover slip using growth factors. You see that the cells that express NLS SOD are much more invasive. And we also used a stem cell reporter that is very efficient in reporting on tumor initiating cell activity. It's basically a construct of the SOX2 OCT4 binding region upstream of either GFP or RFP or mCherry. So what happens is that cells that have activity of OCT4 and SOX2, two very well established stem cell genes, they will glow either green or red, depending on what fluorescent protein used. And then you can just count them by sorting, by putting them under a microscope. In this case, we did flow cytometry, and we saw that cells that had NLS SOD had way more stem cells, SOR6 positive cells in that population than either cells expressing wild type or nuclear excluded SOD. If we injected the cells in mice, and this is a tail vein assay, we just inject the cells through the tail vein. They normally accumulate in the lung. That's where the thinnest capillaries are, so the cells get stuck there. Most will die and get cleared. But so you have your loading 2 hours after injecting the cells. They were luciferase labeled. We injected luciferin, and we saw luminescence coming from the chest. So you see that we loaded, nearly the same amount of cells in all mice. But when we come back 2 months later, we see that only the ones that expressed NLS SOD, the nuclear localized SOD, had recurrences. So these are recurrent tumors, indicating that cells that have nuclear localized SOD, and with that demethylase activity, have way more tumorigenicity and way more capacity to seed metastatic recurrences. We then developed an assay. We got tissue microarrays from patients to look at the frequency of nuclear SOD positive cells in patient tissues. The microscopy core at Northwestern helped us to design algorithms to overlay fluorescence from the nucleus we obtained with DAPI with SOD staining and actually do a 3D reconstruction of those nuclei to be able to quantify SOD that was outside of the nucleus or inside of the nucleus. And by doing that, we could quantify the presence of nuclear SOD positive cancer cells in tumors from ER-PR, which are luminal breast cancers, normally excellent prognosis. ER plus PR, they start to do a basal transition, so they are more aggressive tumors. And ER negative, PR negative tumors, which are hormone negative tumors, they're normally the most aggressive. And we find that the most aggressive tumors, which normally harbor the largest numbers of cancer stem cells, had also the largest number of NSOD, nuclear SOD positive cells in them. We did additional experiments, and this is all submitted for publication. I can tell you that what we found is that nuclear SOD binds to the promoter regions of OCT4 and SOX2 and NANOG, and we also find reduced histone 3K9 methylation on those promoters. So that's what we are proposing as the mechanism by which the methylase activity causes a tumor cell reprogramming to a more tumorigenic phenotype. So why I think this is super cool. As you guys know, the methylation of these histones happen in vivo by several mechanisms catalyzed by LSD1 or by Jumanji-dependent proteins. Those proteins use alpha-ketoglutarate and auction to demethylate. Problem is that in most tumors, auction is not very abundant at the hypoxic cores where stem cells emerge from. And alpha-ketoglutarate is not also very abundant because cells slurp that as fast as they can. So how you get the methylation? Well, we thought that these enzymes are iron-dependent enzymes that actually produce hydrogen peroxide in situ from electrons they use from alpha-ketoglutarate. So maybe iron SOD was using hydrogen peroxide, which is abundant in tumors, to do the methylation reaction. So I think that what this iron SOD is really a alpha-ketoglutarate independent, auction independent demethylase that helps cells to survive the hypoxic environment of tumors and eventually they differentiate to these phenotypes. So finally, I promised you in the beginning that the last piece of this story would be to show you some evidence of the latest kind of work that the lab's doing. So these two previous works, like SOD, changing chromatin organization and gene expression via redox-dependent mechanisms, and nitric oxide affecting how genes are expressed, and FKB target genes are expressed, kind of started to suggest to me, that the changes in the electrochemical state of the nucleus would actually change the cell, would be a major force shaping the phenotypes of the cells, and how I could look at that directly. So that's what this next story is, and it emerged from those two pieces that I showed to you, putting those things together. So to be able to study the redox state of the nucleus directly, I first had to have a sensor. I have to have a way to tell if those nuclei were more oxidizing or less oxidizing. And how did we do that? We used this sensor called ORP1 GFP2 that was developed with Dr. Dyke in Germany, the Cancer Institute, Deutsche Cancer Institute. And what it is basically a GFP that is sensitive to changes in redox and an ORP1 yeast protein. They are connected. So when this sensor is in its reduced form, it's excited at 488 nanometers, and we pseudocolor it green. The response is always in the 530. However, when it's oxidized, it now is excited at 405 nanometers, and we pseudocolor that red. So here's just a validation experiment of cells expressing the nuclear form of the sensor. You see that it's reduced form. You see nuclei glowing green. When we give hydrogen peroxide, we treat the cells with exogenous hydrogen peroxide, pretty concentrated. You see that the fluorescence shifts to red, and we can ratiometrically express this, this difference in oxidation and reduction as a heat map that you see down here. So pretty cool nuclei indicating more reducing, hot nuclei indicating more oxidizing. And what we did is that then, okay, this is great, this is a good sensor, but how we start to get into the physiology of this. So we selected 3 different cell lines, including one that we could transform, and these cell lines were MCF10A, MCF7, and MB231. We have many more now, but let's stick with this 4 at the moment. So what it is MCF10As are regular immortalize but not transform breast, mammary, epithelial cells. So if you implant the cells in a mouse, they do not grow tumors. They behave as normal epithelial cells, and they will eventually die. You see that they have pretty cool nuclei. MCF7 cells are ER positive, PR positive, HER2 negative carcinoma cells. If you implant the cells in mice, they will make tumors, but they don't metastasize. They are not aggressive, and they normally respond to therapy. You see that their nuclei are overall a little bit more oxidizing, but not that much. And you see MB231s. MB231s are triple negative breast cancer cells that were obtained from pleural effusions from a patient. They're extremely tumorigenic, and they metastasize when they're injected in immunocompromised mice. You see that they have the hottest nucleus of all 3 cell lines. We also transformed MCF10A. That was a construct that we got from Kevin Stroll at Harvard. And when we turn on this construct, it transforms those MCF10A cells, remember the ones that were no problem, into a phenotype that is very tumorigenic and very metastatic. And when the cells acquire these more tumorigenic and more metastatic phenotypes, their nuclei also becomes more oxidizing. So there's a clear parallel here between more oxidizing nucleus and more carcinogenic cells. However, this occurs naturally. To be able to study the impact of reactive oxygen species in remodeling the nucleus, I also needed a way to produce reactive oxygen species in the nucleus, and that's how we got this D-amino acid oxidase protein. What it does is that it uses D amino acids. Remember, most our amino acids in our bodies are L. This is the enantiomer, so it's not used to make proteins. That's how we can control the amount of hydrogen peroxide that we produce in the nucleus. And it actually oxidizes these D amino acids that we give to the media and produces hydrogen peroxide wherever we put this protein, whether we put it in the nucleus, in mitochondria, in plasma membrane, you decide, depending on the localization sequence that you tag to it. This is just to show you that when you put D-alanine, you have very hot nuclei. Depending on the concentration, 10 nano, 1 micro, or 10 micro, the cells become oxidizing for longer or shorter periods of time. This is just to show you that there is a concentration dependence here of the sensor, and we can stimulate production of nuclear reactive oxygen species when we use D-alanine. So what happens when we produce reactive oxygen species in the nucleus of the cells? We just turn on hydrogen peroxide by feeding 10 nanomolar alanine. Well, what we saw is that you have an increase in mesenchymal markers from these MCF10A cells that have actually epithelial markers. You see SOX9, you see ZEB1, you see fibronectin, all of them increasing, particularly strong in the case of SOX9. After the alanine is added to the media, we add it for 2 hours and then we wash it away. So you see that in 2 hours, we have SOX9 coming on. It stays for 4 hours. It peaks at 8, and then it comes down. Cells also tend to become more invasive, and they form more colonies, indicating that some cells retain the capacity from this insult to become more tumorigenic. We decided also to get a little bit more encompassing idea of the changes that were happening to do an RNA-seq study. And what we found is that cells stimulated just with 1 pulse of hydrogen peroxide for 2 hours. We analyzed at 4 and 24. You see that there was great separation of the groups that experienced nuclear hydrogen peroxide for 4 hours. At 24 hours, most of the genes normalize, except for some interesting ones that stay on. And when we express that in the form of a heat map, looking at specific genes, you see that 4 hours, a number of genes related to epithelial to mesenchymal transition and wound healing come up, including aldehyde dehydrogenase, SOX9, BAC2. You see here KLF4, which is another one of those stem cell markers. You see PU2F3, which is basically OCT4 coming up. So if you get all of these results and you put in a gene ontology analysis, you see that the pathways that are turned on by a higher oxidation state of the nucleus are involved with epithelial cell differentiation, wound healing, cell adhesion, which are all very important for carcinogenesis. If you remember the first figure that I show you, it kind of also relates to the fact that those more mesenchymal, more aggressive cells, they have more oxidizing nucleus. So that's a clear parallel between that finding and this one. And then we hypothesized that, well, if these genes are coming up, and these are genes that are normally capped in the heterochromatic regions, the cells don't like to express stem cell genes just because that can cause a lot of trouble. Maybe what's going on is that reactive oxygen species are opening heterochromatin and allowing those genes to be expressed. So we did a ATAC-6 study. This is the result. You see that there's an overall pattern of openness once the cells experience this increase in nuclear reactive oxygen species, not only in the close to the TSS region, but across the genome. When we focused on specific genes such as NANOG, SOX9, TWIST, ZEB, all related to EMT, we see there are changes. The regions of these genes become more accessible. So we decided also to do a nuclease assay, focusing on the promoter region of these genes. And what this nuclease assay is, the nuclease will actually chop parts of the gene and that are accessible to the nuclease once you expose chromatin to it. So what we see is that and then we design primers to the promoter regions of these genes. So what you see here is that when we increase reactive oxygen species in the nucleus and we analyze NANOG, promoter, TWIST, promoter, ZEP, promoter, or SOX9 promoter, there is a general pattern of increase after cells start to experience this elevated levels of reactive oxygen species in the nucleus. Some of these genes come on and stay on. Some of the genes come on, peak, and then go down. For instance, NANOG stayed on longer and ZEB1 stayed on longer, but SOX9, you see a peak at 8 that then normalizes, and the same is true for TWIST. But it does indicate that a more oxidizing nuclear state unleashes the capacity, or at least promotes the accessibility of these genes for transcription. And we knew from a paper that had been published a year before in Cancer Cell by Ana Gomez, a very talented postdoc that just got her lab at Moffitt, that this openness of chromatin that unleashes EMT programs is related to a change in the amount of histone 3.1 and 3.3 variants that are deposited into the chromatin. H3.3 is normally associated with open chromatin, and H3.1 normally decorates closed chromatin. So based on that paper, we went to look at H3.1 and H3.3 variants and the impact of reactive oxygen species, both on their expression and their accumulation in the nucleus. These are all isolated nuclei. And what we see is that as cells start to experience an increased level of reactive oxygen species, there is a decrease in the amount of 3.1. So 3.2, those are forms of the histone 3 that decorate closed chromatin, and there is basically no change in H3.3. So that leads to a change in the ratio H3.3, 3.1, with now open chromatin becoming more abundant. We also look at the kinds of genes that would be associated either with 3.3 or 3.1 before and after the alanine treatment or hydrogen peroxide being produced. I'm just showing one analysis here at 4 hours with H3.3 association. And what we see is that when cells are at rest, at basal levels, open chromatin or H3.3 is associated with epithelial cell differentiation genes, with AMPK signaling that is normally tumor suppressive. But after hydrogen peroxide, the regions of chromatin that showed up as open are more associated to cancer pathways such as MAP kinases and GPCRs that get activated. So that indicates that there is a transition from chromatin opening regions that gold genes that are related to EMT and carcinogenesis. But I think the most important experiment that we did, the one that is the one that I show at fundraisers is this one. So basically, these are the same MCF10A cells that you saw before, either untransformed or transformed. When they are transformed, they grow tumors. I told you that. So when we implant it in animals, that's what you see. Tumors start growing and they grow pretty fast over a few weeks. We inject 5 mice with transformed MCF10A cells, and you see that all 5 mice got tumors. So they're very tumorigenic. Now, if you turn on catalase in the nucleus, and catalase is this protein that consumes hydrogen peroxide, it destroys hydrogen peroxide, you see that then when we inject 5 mice with these transformed cells, only 2 get tumors, 3 don't even manifest tumors, And if you look at the growth rate of these two tumors, that's what you see. It's much lower. So it indicates that reactive oxygen species in the nucleus are definitely involved in tumorigenicity and in tumor growth. In these mice, we turn on catalase, these tumors don't go anywhere. So what we are doing now, I think that these three stories, which are just a fraction of the results we have in these areas, really indicate that the changes in the redox state of the nucleus do impact what cells are, what cells become, and what potentials they have for transformation. Either because they impact transcription factors, forming different kinds of complexes. That was an example that came from the NF-kappa-B. Depending on the redox state of the nucleus, whether this redox will impact different regulators. You have the formation of different configurations of the transcription factor with different activities, either because redox process can sculpt the epigenetic landscape, and I think the example was manganese superoxide dismutase becoming iron superoxide dismutase, and now using hydrogen peroxide to demethylate histones and promote gene reactivation. And finally, reactive oxygen species can just change chromatin compaction and chromatin organization, and that will lead to transformation in the transcriptomes and whatever cells can become. We haven't even started looking at link RNAs, nascent RNAs. I think there's another number of layers of how these processes can affect gene expression. I think that redox epigenetics will in time assume the same magnitude of this redox process that have been studied in the cytosol, but I think that it's a much less known field, and actually it's very exciting to be working on it. So of course, it's the culmination of a lot of observations, a work over several years. It had a number of people that participated. I'm mentioning just the 6 that are currently more involved with it and without which the work cannot continue. So thanks to these wonderful investigators. These 2 are my wonderful postdocs. The others are professors I collaborate with. And of course, thanks for our funders, without which we cannot do any work. Thank you for this amazing opportunity to be here in person talking to you, and I will take any questions. Thank you.
Beautiful talk, really exciting and very interesting. In terms of methylation, you saw changes in chromatin methylation. Did you look at any transcription factors or co-regulators, their methylation status?
No, what I didn't talk about is that we are also working with metals, right? And what we see is that cadmium, lead, and arsenic also change the redox state of the nucleus. And one of the genes that is most sensitive to this change of redox state of the nucleus in the case of breast cancer is progesterone receptor. Clinically important for therapy and etc. And one thing that we are seeing is that it also changes the levels of G9A that is associated with the promoter of progesterone receptor. The transcription of progesterone receptor is actually mediated by ER. And the ER G9A duo is what regulates how much transcription you have of that gene. Now, there's a group that described that ER methylation regulates how much it interacts with G9A. So now it became important because of that, because the finding that progesterone receptor is one of these redox sensitive genes in breast cancer, and it's a near target, to determine if the redox state of the nucleus changes the methylation status of ER. And we are pursuing that now. The problem is that there is no ER, MET2 antibodies that are available. So we are trying to immunoprecipitate ER and submit to mass spec and see if we can. Yeah, it's harder, but good point. We haven't done it yet, but I wouldn't be surprised if it's also changed.
I have a question. Very nice, very intriguing seminar. Thank you. So in most of your model system, you try to topically overexpress either SOD nuclear, SOD2, or increase the level of hydrogen peroxide in the nucleus. But I guess from the therapeutic perspective, if those things actually do play roles in tumor growth and metastasis, I think it would be more logical to try to reduce them and see how that does. I was wondering if you've done any type of experiments regarding that.
Absolutely. So let me go piece by piece there. In regards to SOD, we find what triggered this whole research was we started staining because we found that SOD when they overexpressed all the stem cells, etc. And we started just staining SOD in tumors from patients, and we saw that there was a very small number of cells, very rare population, where SOD would very strongly stain the nucleus, and that can always be artifact, but it could be a weird artifact. If it's binding, why is not binding across all cells, but just some? So that's how we started to go about, trying to label it with GFP express, so mutants, where it goes. But the occurrence of nuclear SOD in tumors is real, it's natural, it happens. And it happens more frequently in tumors that tend to be more aggressive. Targeting SOD is very, very hard because, as I told you, it has a very important function as an antioxidant in mitochondria. So if I inhibit it, if I make like an inhibitor or if I knock it out, I have a whole bunch of problems that come in from also targeting the beneficial activity it has in mitochondria. When you knock down SOD in organs or in fetuses, it's perinatally lethal if you do the knockout. And every study that did organ-specific knockouts on all sorts of troubles with those organs. So that I can do. So that's why we switched to understanding what it does in the nucleus. And if I can target instead the substrate, hydrogen peroxide, it uses for its nuclear-specific activity. The easy way to do that is to send catalase there. Catalase is an enzyme that has been studied forever. It's known to detoxify hydrogen peroxide. So to study if that function would help, I would start with catalase. But now you're completely right. How do we do that for therapy? Now, what do you do now? So we have designed a number of catalase mimetics that are targeted to the nucleus. I have just gotten the compound, the first compound I got last week, and we are designing some others with Wuxi in China, and making the compounds to test catalase mimetics in the nucleus. That's how we tend to go about reducing reactive oxygen species in the nucleus for therapy. Now, one thing that we already found that is very interesting. If you get several cancer cell lines, MB221s, MCF7s, BTs, and you express catalase in all of them. For most breast cancer cell lines that we analyze, that has a very powerful anti-proliferative. MB221s, it doesn't work at all. The cells continue to proliferate, etc. I think that the genomic instability there went to a degree that this reactive oxygen species is just something that also happens. Maybe it was important at the beginning, but now it's so transformed that it's past the point where it can use it therapeutically. But for BTs, for MCF7s, for a number of other cells, ovarian cancer cells, pulmonary cancer cells, expressing catalase reduces their aggressiveness, their zinc-1 characteristics, all of that. So I hope that in these cells that expressing catalase works, these therapeutics will also work. One thing that is important is that if you express nuclear catalase into non-transformed cells, it doesn't alter their proliferation, and it doesn't seem to alter some signatures, some gene signatures that they have. So I think that that indicates that these therapies might be more specific to transformed cells that depend on this nuclear redox dependent modeling that normal cells that want to have those oxygens in it. We'll see where that goes. It's very in the beginning. Okay.
You presented very compelling evidence for the case that S-nitrosylation regulates the stability of nuclear P65. The data that you showed us is really all comparing cells with a source of NO to those that don't have a source. And I wonder if you had a chance to look at some more of the physiological regulation, like trans-nitrosylation reaction.
Trans-nitrosylation, no. We went to the point of reconstituting nitric oxide in osmolar knockout mice to see that we get the pro-inflammatory activity back. And using NO scavengers, if you use NO inhibitors, it's a different story. Nitric oxide inhibitors are very difficult to use. They cause a bunch of other problems, and it's very messy. But if you use stuff like CBTIO, that is an NO scavenger, you reduce the expression of a lot of this NF-kappa B pro-inflammatory gene targets in vivo when you activate with LPX. We're doing a few other things too. I mentioned that what nitric oxide is doing physiologically probably oxidants do pathologically. So you also generated the DAO, the NLS-DAO lines. We can use production of reactive oxygen species in the nucleus of the cells that we have. If you do that just before you give LPS or influenza, you see a tremendous upregulation of inflammatory cytokines in these mice. So it indicates to me that either nitric oxide doing this or hydrogen peroxide mimicking nitric oxide in the nucleus promotes pro-inflammatory gene expression. Where we like to take this is in the context of aging. I think that one major thing that may be happening is that because of mitochondrial dysfunction with age, you have an intrinsically more oxidizing nucleus of immune cells of older people compared to younger people. So once NO displaces SOX from the inflammatory region, they are not free to be expressed, but to stop the expression of those genes, you need to reload SOX, and oxidants won't allow it to happen. Because it oxidizes those diols, we have described in the JN paper that are two cysteine residues that are critical for SOX function as a disease scavenger. That's cysteine 147 and cysteine 179. Of the 5 cysteines that the SOX has, those are the two that are used. I think they will be also oxidized. And when they are oxidized, you cannot reload it from those genes. So once those genes are activated, they are activated for much longer, and that's why you have a much more robust inflammation in patients that are older. It doesn't translate though into viral clearance, because I see that younger and older mice clear influenza virus with the same efficiency mostly, but the inflammation that you get from the older or mice that have experienced this reactive oxygen species shock in the nucleus is much higher than in the younger non-hydrogen peroxide mice. So it almost indicates to me that what's happening in older people in these cases is they have much more inflammation per unit of viral clearance. And that leads to all those troubles and non-fibrosis and lung inflammation and ultimately leads to poorer outcomes.
Anything from the online audience?
No, you is here. Hi, thank you for a wonderful talk. I'm an epidemiologist, so sometimes I feel like I don't know how to talk this language, but I'm curious about two things, two unrelated questions. One, like, what would environmental triggers of this nuclear reactive oxidation, and like, is it dependent on the cytoplasmic environment at all, or
it does. I see what you're saying, and then I didn't talk about that. Maybe we should save it for another opportunity, so I have to be brought back. Okay. So no, it's perfect. So one thing that we saw, remember that I just talked about the metals work we have. So I believe that any mitochondrial poison, for instance, herbicides, tar, quad, the Parkinson, you've seen one very popular, rotenone, glyphosate, all of these, and metals have the ability, metals I'm sure, is cadmium, arsenic, and that is really external. They do increase the levels of reactive oxygen species. And I think that's because they increase the levels of reactive oxygen species by more. There was a paper in Scientific Advances last year that showed that mitochondria and the nucleus are connected to channels, and a lot of things diffuses to these channels, including reactive oxygen species. Now, if you increase the production of reactive oxygen species here tenfold, they will flow more to the nucleus, and that's how we maintain that level of higher oxidation. In the case of breast cancer, progesterone receptor is a redox regulated gene. We see that when we treat cells with macros, progesterone receptor is downregulated tremendously. The function of progesterone receptor is downregulated tremendously, and those cells individually become more aggressive. How now if you use nuclear catalase, reverse all that, if you put progesterone receptor back, reverse all that. Now we actually did the immunology of that. Well, since there's some evidence, I don't know how much, and that's why I need to collaborate with the immunologists, that exposure to air pollution kind of people that are exposed to air pollution disproportionately get more progesterone receptor negative breast cancers that have a way poorer prognosis. I think that what really motivates me to do this research is that, eventually you'd like to have cleaner air for everyone, eventually you'd like to move people out of highly polluted zones, humanly, politically, economically, that's not possible, right? I don't see in the next 30, 40 years, we extinguish poverty around the world and everybody can be nice places. So how do you immediately get to that problem? Well, if we are successful into developing this nuclear targeting antioxidants, and we see that at least in cells using catalysts, you restore progesterone receptor expression in those cells that have been poisoned with metals, and more. You restore the response to tamoxifen. Then you can give this chance when they go to treatment. Because now we have a pharmacological solution for environmental things, right? More step to the students earlier. Tamoxifen is not the best thing to have to treat breast cancer in this country, but it's a first-line chemotherapeutic that is normally the only alternative many women have in poor that also suffer from pollution. Once tamoxifen is not working for them, it's pollution. So, restoring response to tamoxifen is very important, particularly in the context of social inequities and pollution. But to delve deeper into the geography and the epidemiology of things, I don't have the expertise, but I believe from the little that I understand in this area, that there is some indication that women that are exposed to pollution, particularly by heavy metals, have a prevalence of PR-negative tumors that is not explained by genetics. And I think that one study that was particularly interesting that I read is that Bangladesh women living there, they have an incidence of PR-negative tumors of 27%, something like that. That's very high. Normally, in this country, it's 12, 13%. There's 27. But they also studied immigrants that left Bangladesh to live in London. There's a cohort in London and a cohort in California. In those women, the incidence of PR-negative tumors are somewhere between 16 and 19%. Much lower than what it is in Bangladesh. I know that this population is meant to intermarry, so I don't think it's genetics. I think it's environment. And one of the deficiencies of the studies is that they looked at immigrants. They didn't look at people that left when they were 2-year-olds or that left yesterday. So maybe if you stratify the population, you see that removal earlier would actually lower the proportion, even closer to what is right now. But the studies just look at people that left.
I'll share some other studies with you. I do think this is really interesting that I think the field of like breast cancer epidemiology has historically focused on what are risk factors for ER positive disease just because of the sample size. It's nice to see an example where there are strong differences in mechanisms, right? Estrogen status and that.
And ER positive is deceiving. I came to learn after working in this field for a long time that ER is very deceiving. I have a person in the group that is an expert in ER signaling for that reason. Because sometimes, you see a decrease in 5% of ER expression. You have, let's say, whatever number, 500 units of staining, whatever, and 480, right? So you would say ER positive. But you don't have the function, because those are PR negative. Why I like PR? PR is a target of ER, and if ER is not functioning, you don't get ER. So sometimes you see very little ER decrease in terms of protein levels, but PR is gone, which means that you have PR but it's not functional. Sometimes you see ER going down by 95%. You have like a very black band, very strong, like a 91, but ER is functional. Then you have the function. The function is the same as what you have when you have 100%. So that's why it's deceiving. I just wonder how many tumors go to pathology and come back here positive. You say, okay, that's here positive tumors. But they aren't. Because function and staining in this case is very different. Like these cells, for instance, that we treat, depending on the cell line, you give to mouse, you lose anywhere between half to 95% of the ER. And you lose between 5% and 100% of the ER. So really, that's why ER without PR in my mind don't go together, because one is amount and the other is function.